We now come to chapter 26, looking at the urinary system. So if we look what the urinary system does, of course, we think about urine, which is our main excretory system. So we're trying to get rid of things at this point. Of course, we have a couple kidneys that are used to do that. The ureters help connect the bladder to the urethra to actually be emptied. So the kidneys are kind of our filtering system. So they're filtering all the fluid, all the blood that's coming in. And then any of the waste would be removed into the urine. So it consists of excess water primarily, but also excess ions that are there, waste products like urea from the breakdown of proteins, as well as anything that may be toxic to the body. So they also have, though, other activities, but the main function of the urinary system is to get rid and filter out waste. So again, main function here is excreting waste. However, it's very important in regulating our blood volume and pressure. So that's why a lot of times whenever somebody has high blood pressure, they're given a diuretic because our kidneys are helping regulate that blood volume, which then in turn regulates blood pressure. Also regulating solute concentration, so looking at ions like sodium chloride, potassium, calcium, and others as well, including urea, which again comes from that breakdown of proteins as we digest them. And because of the hydrogen ions, it's also important in playing a role in pH. It actually helps regulate red blood cell synthesis by secreting. So again, thinking about not only removing things, but the kidneys actually make a protein called erythropoietin, which is what makes our red blood cells. And it's involved in regulating the synthesis of vitamin D, and vitamin D impacts our calcium levels. So again, waste and excretion main function, but there are other functions as well. All right, so if we look at some of the anatomy, of course, the kidneys are kind of a kidney bean because they kind of are bean shaped, but they have retroperitoneal, which is behind the peritoneum, or in the non-mesenteric region there of the abdomen. So posterior abdominal wall, right there at the lumbar vertebrae and the rib cage, and the right kidney just a little bit lower than the left. Looking at the external anatomy of the kidneys, there's the renal capsule, which is made up of fibrous connective tissue that surrounds the kidney. There's some fat tissue or adipose tissue that engulfs and kind of surrounds that renal capsule, and it helps provide cushioning to protect for impact whenever we have impact, to protect the kidneys. The renal fascia, that's just this thin layer of loose connective tissue. It's involved in anchoring the kidneys to the adipose and the abdominal wall to keep them in place. And then finally the hilum, which is the renal artery and nerves where they enter, and then the renal vein and ureter exit through this hilum. So that's where the nerves and the blood supply are coming in, and it opens into what's called the renal sinus. Right, so again, just looking here, of course, our left and our right kidneys, the hilum as it shows here, that's where, again, the nerve and then the arteries and veins are coming into the kidneys on both sides. And then if we look at, again, kind of the upper view here looking down, again, we can see that renal capsule, which is the outer layer, but then it's surrounded by that adipose tissue. And then the renal fascia is what actually connects it to the fat tissue and the surrounding wall. Now, if we look at the internal part of the kidneys, the cortex is the outer area that would be up to that capsule. And it's made up of renal columns that are part of cortical tissue, and they go into what's called the medulla. And the medulla is the inner area, surrounds that renal sinus. Within the medulla are the renal pyramids, which have a cone shape, hence the name. And the base projects into the cortex is what are called medullary rays. And then the apex of the, which is the tip of the pyramid, is the renal papilla, which points toward that sinus. There are two calyces, the minor calyx, which is the papilla that extend out of them into a funnel, and then in the major, they converge to form what's called the renal pelvis. And that renal pelvis is an enlarged chamber that's formed by that major calyx, and then ultimately the ureter exits at the hilum and connects it to the bladder. Looking here now at the inner part of the kidney, so again we have these renal pyramids that are shown here. The two main parts, so we have the renal medulla that's on, switch my color. So we have the renal medulla on the inside, the renal cortex here on the outside. So this portion here is the renal cortex with the medulla on the inside, and that's making up those pyramids. Then the calyces, so we have again the major and the minor calyces ultimately coming out into the hilum, which is where that ureter is going to connect into the bladder and then ultimately out through the urethra. All right, so now talking about a nephron, which is kind of a functional unit of the kidney, and there are a few different parts to the nephron. We start with what's called the renal corpuscle, then we go to the proximal convoluted tubule, the nephron loop, more commonly known as the loop of Henle, and then the distal convoluted tube. So we have renal corpuscle, proximal convoluted, loop of Henle, distal convoluted, and this is the path the urine is going to flow through. So it's going to start at the distal convoluted tubule, goes through what's called the collecting duct, then it moves toward that renal papilla to the papillary duct, then to that minor calyx, and then it continues on from there. There are a couple different types of nephrons, and that includes the juxtamedullary nephron. This is in the renal corpuscle near the cortical medullary border, and these loops extend deep within the medulla. And this is about 15% of the nephrons. The main ones are the cortical nephrons, and this is where that corpuscle is near to the periphery of the cortex, and the loops do not extend very deep into the medulla. So that filtration part is called the renal corpuscle. So that's what we're talking about, what's filtering out all of the fluids and the ions and solutes. All right, so if we look at the kind of the flow and everything through this, so again, this is our renal pyramid. So again, we have our cortex on the outside, our renal pyramid that's part of the medulla on the inside, and then at the very apex here of our of the renal pyramid, we have that renal papilla, or that, again, that apex end, and that's where we're going into that minor calyx. So if we kind of look at the flow through this filtration, so we have the renal corpuscle. Again, this is kind of the main filtering unit to kind of get us started. So urine's going to flow through the renal corpuscle. It's going to go through the proximal convoluted tubule, and then it's going to come down into this loop of Henle, hence the name, because it looks like a big loop. And then we come back out into the cortex, and this is where that distal convoluted tubule is. And then it exits into that collecting duct, and then the collecting duct goes down into the minor calyx and then continues on from there. So again, the two different types of nephrons differ by how much they go. So again, this one is going deep into the renal medulla, whereas the other is shorter. It's not quite as deep into it. So they both have the same parts. It's just how long or how far into the renal pyramid that loop of Henle goes. Now, the renal corpuscle is also, and more commonly known as Bowman's capsule. So this is that outer parietal and visceral region. So the parietal portion contains simple squamous epithelium, and then the visceral make up what are called podocytes, or the cells that make this up. And then what's called the glomerulus is a network of capillaries, and the blood is entering through the afferent arteriole and leaves through the efferent arteriole. So again, our renal corpuscle here, or Bowman's capsule, comes in through here. So this entire thing, again, known as Bowman's capsule. And then that glomerulus, which is showing down in there, is this connection, or this collection or network of all of these capillaries that are inside of that glomerulus. All right, so if we look at this capsule again, we have the two layers. So we have the parietal layer, which is the outer layer made up of those simple squamous, actually becomes the cube shaped where the Bowman's capsule ends, and that proximal tubule is going to begin. And then the visceral layer is the inner portion made up of those specialized podocytes that wrap around those capillaries. So again, here's our proximal or parietal layer here on the outside, and then that visceral layer inside made up of those podocytes that are circling or encompassing those arterioles. So you can see them a little bit better lining that here. Now we have what are called fenestrae, and these are kind of window-like openings in the endothelium of those capillaries. And then filtration slits are gaps between those podocytes, and the basement membranes is sandwiched between the endothelial cells and those glomerular capillaries of the podocytes. So again, we have the podocyte here, that visceral layer of the glomerulus is here on the outside. And then we have these various slits that you can see here, and then the fenestrae, which again are going to be these little openings that you see. Let me just switch my highlighter there. So again, these little openings here are called fenestrae, and then the filtration slits are also present to allow for circulation and filtration. So if we look at this filtration membrane, again, we have endothelium, just like we do on our vascular tissue, that basement membrane, and then the podocytes themselves. So the first stage of the formation of urine occurs here in this filtration membrane. So this is filtering the fluid from the blood of the capillaries and is going to move it to the inside of Bowman's capsule. So the characteristics here for that filtration, so we have porous capillaries. So again, those fenestrae, those kind of openings there, are very permeable, meaning lots of things can come in and out of the glomerular capillaries. So there are large proteins, blood cells, though, cannot pass through. So they're bigger openings, but they're not huge. They're not letting anything in, but definitely ions and a lot of solutes, just not really big proteins or cells themselves. And then they also have a porous visceral layer. So again, those filtration slits that are not, so the fenestrae are on the internal part there, and then that visceral layer that wraps around those capillaries have the filtration slits that are allowing that movement into the capsule. And there's a lot of pressure going through here, so everything's moving from a high to low pressure gradient. So those capillaries, glomerular capillaries, have a higher pressure because the efferent arterioles, the ones leaving, have the smallest diameter. So they control that pressure by using diameter. So the glomerular capillaries have that higher pressure, again, because there's a smaller diameter in the efferent than in the afferent arterioles. Now, the juxtaglomerular apparatus here is a specialized structure close to the glomerulus, hence the name. This is where renin gets produced, which is one of the things that controls filtration and fluid retention. So juxtaglomerular cells here are a ring of smooth muscle in the afferent arteriole, where the latter enters the glomerular capsule. And then the macula densa, these are specialized tubule cells of the distal convoluted tubes, and then that lies between the afferent and efferent arterioles. All right, then as we move into the tubules, so we have our proximal convoluted tubule. It has simple cuboidal cells with lots of microvilli. So just like with our digestive system, those microvilli increase the surface area to allow for better or more surface area to allow for filtration in this case instead of absorption like we had in the digestive system. And then with the nephron loop or the loop of Henle, there's the descending limb, which would go down. So that's the first part of that tubule. And then we have squamous epithelial that become thinner as we move through the descending limb. And then it loops, and then we move into the ascending limb. So the first part is a simple squamous, and then as we move, they become thicker cells and become simple cuboidal. By the time we get to the distal convoluted tube, this is shorter than that proximal, but both still have the simple cuboidal. And these cells, though, are smaller and have very few microvilli, because at this point we're kind of to the end of that filtration unit, so we're not doing as much filtering. That's more at the beginning. And then we move into those collecting ducts, and that forms where lots of those tubules come together. Again, larger in diameter, also simple cuboidal, so that's kind of easy to remember. The majority parts of our nephron are simple cuboidal. So it's just remembering that the descending limb and the beginning of the ascending limb have those simple squamous. The rest of it is simple cuboidal. And then from those medullary rays, it leads to the tip of that renal pyramid. So again, just kind of looking at our different cells here. So as we move into the proximal convoluted tubule, again, we have the cuboidal cells that you can see here. Again, lots of these microvilli to increase that surface area there for more filtration to occur. Those are going to continue through the first part of that descending limb of the descending loop of Henle. Then as we get down toward the bottom of that and beginning up the ascending loop, now we're going to have these more simple squamous cells. They're going to start flattening out. Then as we move up that ascending limb and then into the distal convoluted tubule, now we're back to those simple cuboidal cells. But again, fewer microvilli there than what we have with the proximal tubules. So now if we look at the actual production of the neuron, again, nephrons are that functional unit that regulate body fluid. The three main steps of urine formation, first is filtration, then we have the tubular reabsorption, and then tubular secretion. So if we look at this filtration part here, so again, blood pressure in those capillaries forces the fluid and small molecules out of the blood. So now it's called the filtrate. And it's non-selective at this point. It just separates based on size or charge of the molecule. So the smaller things are, the faster they come out. If they're really big, they can't get out. So again, it's the filtration's comparable to kind of emptying the junk drawer with everything except for your big permanent kind of items. So it does not remove everything in the blood. Again, bigger proteins, cells, things like that are going to remain. So it removes only substances small enough to go through that filtration membrane. So the fenestrae or the filtration slits. Right, now as we go into the tubular reabsorption, now the cells of the renal tubules have a lot of different transport proteins. And again, those are the kind of the channels that allow hydrophilic molecules to move across that phospholipid bilayer. So they're going to transport or move water and some of the filtered molecules from the filtrate back into the blood through what are called peritubular capillaries. This prevents them from being lost. So again, it's kind of recycling them as it moves through. So it's going to get filtered during the filtration, but then it gets put back into the blood. So again, it's kind of saving the items from our junk drawer. So most of the filtered water and useful solutes get returned to the blood by the time the filtrate is modified to urine. Everything else, though, the waste and the excess, that's going to go through the urine. And then finally, we have tubular secretion. So some of those tubule cells actually transport additional solutes from the blood into the filtrate. So some of these may not have been filtered by that filtration membrane, so they actually move more through those transport proteins, so they're transported or secreted out. So they weren't filtered to begin with, so then of course they weren't reabsorbed, but they do get secreted before urine is released. So again, kind of back to our junk drawer, this would be the throwaway items, made it through, but now we're getting rid of them to be removed in the urine. So just looking a little bit more at that filtration, again, movement of the fluid, it's coming from the blood through that glomerulus or Bowman's capsule, through that filtration membrane. Again, the term, once it comes out, is now called the filtrate, which includes the small molecules, of course, ions that are very small, that wouldn't normally move through the phospholipid bilayer, but they can move through the fenestra or those filtration slits. Again, the pressure difference is what forces them through the membrane, going from higher to lower pressure. And then we have what's called the renal fraction. So this is the part of our total cardiac output that passes through the kidneys. So it varies, but again, how much of that what's coming in the blood actually gets passed through the kidneys. The renal blood flow rate, this is the rate that the whole blood flows, so how long it takes all of the blood to flow through the kidneys. So that's determined by the cardiac output times that renal fraction. Again, there's an average of about 1,000 milliliters per minute, just to give you an idea. And then we also have the renal plasma flow rate, and this is the renal blood flow rate that we just mentioned times the fraction of blood that's in the plasma, and that's about half of what the renal blood flow rate is. So that glomerular filtration rate, the GFR, this is the amount filtered each minute. So just to give you an idea, it's about 180 liters a day. But it's determined by that renal plasma flow rate times the filtration fraction. But again, it's just the amount of filtrate per minute that's moving through the kidneys, and then that filtration fraction is the part of the plasma that's filtered actually into the glomerular capsules. And then just to put some kind of numbers here, so the average urine production each day is about 1 to 2 liters, but most of the filtrate gets reabsorbed. So lots of blood is coming into the kidneys, but of all the blood that passes through the kidneys throughout the day, only about 1 liter or 2 of that filtration is going to be removed. And that filtration membrane, again, just that barrier here to prevent the blood cells and some of those larger proteins from going into the Bowman's capsule or glomerular capsule. But again, much more permeable than most capillaries because of the fenestrated capillaries. Again, those little openings in the capillaries. Again, our basement membrane there, and then the podocytes of the visceral have those filtration slits as well. So some albumin and small proteins, though, can enter the filtrate, but then they get reabsorbed and then metabolized in the proximal convoluted tubules. So very little protein is typically found in the urine. So the presence of urine or protein in the urine indicates some type of problem or some type of condition.